Welcome, everyone, to the 2025 Gottfried Schmier Memorial Lecture. I am so happy to see all of you because as long as we have been doing this lecture, we have mostly done them virtually. Only this one and maybe the first one were maybe there was one other one that were in person. But first, I want to, my name is Sean Murphy, and I am here in UW DLMP and at Seattle Children's. And Dr. Schmier was a great friend and mentor of me. So before I introduce today's speaker, I want to remember Dr. Schmier for a few minutes. He led a really interesting life, one which says you can kind of reinvent yourself at any point in your life if you want to do something different. And he spent the first very impactful part of his life as a coagulation expert. He trained in medicine in Austria, studied biochemistry at the University of Vienna, and then came here in 1967 to work with Earl Davy and Hans Neurath, where he did a lot of impactful things, including isolating von Willebrand's factor and the factor VIII complex, which is shown, the publication is shown here. And he was recruited to join then laboratory medicine by our founder, Dr. Strangerd, and he served as the chief of clinical coagulation until 1998. And then in 1998, when he quote unquote retired, he decided that he would go and spend his time working in the tropics and studying tropical medicine and microbiology. And to do that, he went to the Gorgas course, a famous course in clinical tropical medicine, where he was most certainly the oldest person in the course. You see him circled here. And then for the next many years, he spent his time between here and South America. He got a master's in public health and tropical medicine at the famous Tulane course. He spent time in Lima, Peru, and he spent time in the Amazon with his friend who studied leprosy, Sineso Talahari. And during this time, he went up and down the Rio Negro and the Rio Amazonas. He told lots of stories, took care of a lot of patients, really saw a lot of diseases that we only see in textbooks firsthand, and brought a lot of that knowledge back here where he worked in the leprosy clinic. You may remember he rode his bike to work into his 80s. I don't ride my bike to work at all, so I have a lot of work cut out for me, and he spoke so many different languages. He was a great friend to many of us and a great teacher, and this is just a picture of him in the clinical microbiology lab teaching, not only people in our department, but ID fellows like our guest today, and really a great friend. So I'm honored, I'm grateful to Dr. Baird that we're able to host this every year. And with that, I'd like to introduce this year's esteemed speaker. So we are joined today by Dr. Audrey Odom-John, who's an MD PhD and is the chair of pediatric infectious diseases and the chief of PedsID at CHOP in Philadelphia. Dr. Odom-John actually did her fellowship here at the University of Washington in PedsID, and after that, she went to WashU, and then after that, she ended up out in Pennsylvania at CHOP. She has been interested in malaria, in a variety of different metabolic aspects of malaria, and today we'll hear about something that dovetails with our diagnostic interests a little bit, which is called Breathomics. I don't know the full scale and scope of what you're going to talk about. And she also has another interesting part about her appointment in Pennsylvania. She's part of not just the medical school, but the veterinary school. And I think, as I've discovered today, this gives her a really unique opportunity to explore and have overlapping interests across all of that. And so, you can ask her about that at some point if you get to meet her here or afterwards. But thank you very much for joining us, and we look forward to your talk.
Thank you so much for the kind introduction, and thank you so much for inviting me to be here. I will say yes every single time that I get invited to come back to Seattle and UW, and it's just been an absolute pleasure. I also knew to ask to come in June, and the weather has indeed served me up nicely. So it's just been an absolute delight to get to meet with old friends and to meet some new folks as well. So I'm going to tell you a little bit about how we ended up in this crazy field of breathomics, which really started from work that we did on the very basic fundamental science stage in the lab, and then has gone all sorts of crazy directions since then. So it's quite the science journey, and I hope you are buckled up. No relevant disclosures for this one. So the journey really starts with an interest in malaria. And, as a pediatric infectious disease physician, malaria is, of course, one of the most important global causes of illness and death in children. In 2021, there were almost 250 million cases of malaria worldwide, over 500,000 deaths. And really, almost all the deaths are concentrated in children under the age of 5 and in pregnant people. As I mentioned, it's a major cause of death globally in children under the age of 5, and it's really, one of a lot of infectious diseases that account for a proportion of deaths in children. But if you look at those causes of death, many of which still are shocking to me, for instance, the number of children who die of measles, which is completely vaccine preventable. The top causes there, pneumonia and diarrhea, really have a lot of different pathogens that cause those things. But the deaths due to malaria really due to infection with a single species of a single protozoan parasite. And we have made some really great progress in reducing the number of malaria deaths worldwide over the last 20 years. But since the pandemic, we've been going in the wrong direction. So there had kind of been a plateau, and now things are not looking so great. So cases are on the rise and deaths are on the rise, and we clearly need to sort of redouble our efforts. And that's particularly challenging in 2025, as it turns out. And part of that is because malaria is concentrated in the parts of the globe that have the least amount of money, and so are really quite dependent on the resources of others to really fight this disease. As I mentioned, malaria is due to infection with a protozoan parasite, and there are a number of species that cause infection in humans, 5 predominant ones, and I know this group knows it well, but just to remind you, this is an infection that infects red cells, and you can see the parasite looking pretty in human red cells here in sort of their classic signet ring and applique forms. It's really quite a beautiful infection, unfortunately. This is a disease that's that's transmitted through the bite of the mosquito. So I know that folks who are not parasitologists glaze their eyes over every time they see a life cycle, but I won't go through too many of the details, but just to remind you that this is a mosquito-borne infection. There's this asymptomatic stage of infection in the liver stage, and then there's this cycle of growth and development that takes place in the red cells, and that can happen over and over and over. And that asexual replication in the red cells is really what causes the signs and symptoms we associate with malaria. And of course, that's the stage we need to be able to detect in order to diagnose malaria infection. Malaria is still present in the US. Although it used to be endemic here, about 2,000 cases of malaria are still diagnosed each year in the US. And as some of you may be aware from recent news reports, there were even octonus cases in the U.S. In 2023. So poor Florida and Texas, which have other problems as well, but they also had circulating cases of Plasmodium infection. And my former graduate student, I say former because she got her PhD like 2 weeks ago, Brianne, did a certificate of public health while she was in my lab. And as part of that, worked with one of our Peds ID fellows, Seish Sundararaman, to actually do a retrospective review of what pediatric malaria looks like in the U.S. And I'm not going to go through all of this because I want to get to some of our basic science work. But I just bring it up because it really highlights actually some of the problems of malaria worldwide. So we actually included 9 major pediatric hospitals around the country, which, as it turns out, included Seattle Children's. And we looked at 171 cases of pediatric malaria over an 8-year period, 32% of which actually met CDC criteria for severe malaria. And there were a few things that came out of that were really quite striking, one of which is that co-travelers are at risk. So almost a third of the children that we diagnosed with malaria had traveled with someone else who had malaria. And at our institution alone, we had 7 separate groups of siblings among malaria patients. So 6 of 7 groups were treated after the same trip. One had a kid who got malaria again the next year as well. So we've actually changed some of our protocols to screen co-travelers. But really what I wanted to highlight are what we saw in these groups as the symptoms of pediatric malaria. And as you will see here in this sort of symptom map, it's all over the place. So the predominant symptom of pediatric malaria is fever, right? 93% of our patients had fever. The ones that didn't, I think, were the asymptomatic siblings that got screened. And then all the other symptoms are really, there's tummy ache, there's diarrhea, there's cough and runny nose, there's body aches, there's chills, there's headache. It's really not very, very nonspecific. And as a result, some of our patients got missed because it's not obvious what you have when you present with malaria. And in fact, in these 151 cases of pediatric malaria in the US, 28% of them had a previous encounter with the healthcare provider where their malaria diagnosis was missed, giving them an odds ratio of about 4.0 for ultimately presenting with severe malaria. So it turns out to not be good to miss malaria infection, and a lot of that happened even in the emergency room when their parents brought them in. And that really highlights the major challenge, both with managing malaria in the US and managing the malaria globally, and also eliminating malaria, which is that essentially every single patient or every single child who has suspected malaria in an endemic area should be tested. And I don't know how many of you are parents and have, say, parented a 2-year-old. Your average 2-year-old in anywhere has approximately 8 to 10 febrile illnesses a year. And the predominant symptom is fever, right? So essentially, your average 2-year-old who lives in a malaria endemic area, which is, let's just point out, half the world's children, should be tested for malaria roughly 10 times a year. And that's if you're doing it the right way, right? So this is a lot of testing. There's no place that's actually testing every single kid who has a fever. So we clearly need additional access to testing, and we're gonna have to do a lot of it. And then, of course, everybody who gets diagnosed gets treated, and then we need to track all those patients. But testing is a major, major important point in both providing the adequate clinical care and then ultimately tracking and doing malaria control measures appropriately. And so I know that Sean will have talked to you all a lot about diagnostics for malaria, so I won't go into this in too much detail, but in most places, they don't have access to fancy PCR abilities, and most places will diagnose malaria with either classic blood smears, smear it out on a slide, stain it, look at it, or rapid diagnostic tests, these lateral flow-based RDTs. And the RDTs have been great. They've been transformative. They detect a really phenomenal antigen called HRP2, and this protein, HRP2, is actually produced in large quantities by the parasite. It's exported out of the parasite, so it's just in the bloodstream, regardless of where the parasites are in the body, and that's great. And so you can detect it just like you might any other antigen bilateral flow. But as we all know, nothing in biology makes sense except in the light of evolution. Or put another way, life finds a way, and if you roll out enough HRP2-based rapid diagnostic tests and you only treat the people who are HRP2 positive, lo and behold, you get emergence of parasites that don't need to make this non-essential protein anymore. And so there are parts of the world, Ethiopia and Eritrea, where up to 80% of parasites can no longer be detected by the rapid diagnostic tests. And there's pretty good evidence that this is on the rise. I don't need to explain to you all how not making the protein means you have false negative testing. But there have been modeling efforts that, and this was pre-pandemic, so the rate here has been slowed a little bit, that the current RDTs may stop working by and large across Africa by 2030, which seems very close to now. So there's a clear urgent need both for access to malaria testing, but also probably for some new antimalarial testing that malaria testing that doesn't rely on HRP2. So my lab's approach has been very simple and relies on this fundamental property of people, which is that they stink. And what we mean by that is that people give off small, generally nonpolar organic molecules that we detect, right, with the odorant receptors in our noses as characteristic odors associated with people, but that you can also detect other ways, like mass spectrometry. And we've been interested in human odors as a source of information about health and disease, really because of all the information it gives. So if you just look at breath, for instance, there are over 400 to 600 different volatile organic compounds that have been described in normal human breath. And there's really a rich spectrum of stereochemistry and information about the underlying biology that you can get from those VOCs. And we got into this really from the basic science side of things. I had an undergraduate in the lab who got really fascinated by some observations that were in the literature that mosquitoes seem to smell who has malaria. So this is just one of those studies where they actually put children in boxes and then dropped mosquitoes to see which direction they would go. And it turns out the mosquitoes were more likely to go after the kids before they got treated for their malaria than after they were treated for their malaria, and particularly children that had the sexual stage of the parasite that's taken up by the mosquito, the gametocytes. But this is just one of those studies. There have been studies in adults, there have been studies in mice, and there have even been studies in avian malaria, bird malaria, that mosquitoes are more likely to be attracted to or to bite individuals who are Plasmodium infected. And so that suggested to us that we would actually be able to see different odors in cultured parasites. And so the student who was in my lab, Megan Kelly, who's now a junior investigator at Scripps Institute, figured out a way to actually analyze the odor in the headspace over cultured parasites. So we can grow malaria parasites in human red blood cell culture and actually collect and analyze the compounds over those. And we were able to identify a group of compounds called terpenes that are not characteristic of human-produced compounds. They look a lot like the sort of things that plants make. And we found them enriched in cultures of infected red cells and were not present in uninfected red cells. And so we wondered whether or not these compounds might actually be compounds that could be detected by malaria transmitting mosquitoes, the Anopheles mosquitoes. So we partnered with mosquito odorant receptor biologist John Carlson at Yale, and they have a system where they can put odorant receptors of the mosquito in fruit flies so that they can actually figure out one at a time, so they can figure out what these odorant receptors respond to. And lo and behold, mosquitoes can actually smell the compounds that the malaria parasite was producing. So we were able to identify, for instance, this odorant receptor 21, which recognized a compound pinene, and odorant receptor 75, which recognizes a compound limonene. So really suggesting that mosquitoes have the biochemical machinery to actually detect these compounds that the parasite is producing. And in work that I wish I had done but did not, there have been subsequent studies to show that actually the mosquito is indeed attracted to malaria parasites in culture. So this isn't just something that happens in people. They are actually attracted to cultured parasites, and in fact in sort of a dose response sort of way. So this is all kind of crazy and suggests the malaria parasite is somehow tweaking mosquito odor attraction in order to increase the likelihood of transmission. But as I began to talk about this work many years ago, in forums just like this, the thing that came up over and over is people would ask me, okay, this is really cool, and it looks like it's happening in culture, but is it actually relevant to human infection? Do kids with malaria actually smell different? And does the mosquitoes actually care? And so this was challenging, as I was like a malaria biochemist, and I did not know how to know whether or not kids smell different. But we began to dig into this literature on how you actually analyze human odors. And so I partnered with Indy Trehan, who was at WashU with me at the same time and now is here at Seattle Children's, and Chad Schaber was the graduate student in the lab who did this work. And so we did a pilot study in Lilongwe, Malawi, on children with and without malaria who were presenting with fever. And we developed a system to collect and analyze breath. So we have essentially a setup where the child will blow into a bag. It looks like a plastic bag, but they're essentially low VOC, so you don't detect all the VOCs from the plastic on the columns. And essentially you fill up the bag, and then we pump that over small thermal desorption columns that we can ship back to the US for analysis. And you can just see the difference between, say, a room air control and a pediatric breath control, and this is just analyzed by GCMS. This may or may not be a small human genetically related to me who is much larger now, and that may or may not be my kitchen. But you can see in the inset there, isoprene is a compound that is known to be enriched in the breath of people, and you can see that my daughter's breath was in fact enriched in isoprene, as you would expect. And so our biomarker discovery pipeline is similar to others. You get your sample from patients with and without your disease of interest. We do mass spec, and then we have a variety of different statistical tools that we use to look at discriminatory volatiles in the different groups. And this is just one of the children enrolled in our initial study. And you can see we can get reliable breath samples from children down to about four and up for sure. 3-year-olds are a little iffy. Girls are better than boys, I'll just say. But this initial pilot study allowed us to identify some discriminatory volatiles in children with falciparum malaria compared to children who otherwise clinically were indistinguishable. And we were able to use a cumulative abundance metric to give us some information about which children did or didn't have malaria. Obviously, it's not got the diagnostic performance characteristics you would like for a clinical diagnostic, but it really, in this pilot study, gave us a lot of hope that there was some information to be had in the breath samples. So we've subsequently gone on to do some independent validation, and this work has been done in partnership with Carl Seidel at the Blantyre Malaria Project. And here we not only enrolled febrile children, but enrolled many healthy siblings that had been brought to the clinic with them. And in addition to collecting samples at the time of their initial appointment, if children were positive by RDT on their initial appointment, we had them return 3 days later and collect samples so that we could begin to see how the breath volatiles actually change during the course of treatment. And we had a really nice study population. We had a range of parasite densities because one of the things that we hoped to do was to identify whether our biomarkers were more or less sensitive in patients that had low density parasite infections. And an important thing that we have learned to do now is a 24-hour dietary recall because sometimes things that you eat can really be clearly detected in your breath, as evidenced by one of our studies in the US where a child ate a piece of lemon cake an hour before his appointment and his limonene levels were literally through the roof. So now we know to check. And fortunately, our groups were similar. And what you can see in this sort of dimensionality reduction, where you just look at an overall profile of VOCs, is that children with falciparum malaria look very different than children who have fever from other reasons, and they all look very, very different than the healthy siblings. So the healthy siblings are in green there. They're clearly distinct from the children who come in with fever and who are sick. And what we see is that the children with malaria cluster really tightly, whereas the children who have febrile illnesses, obviously a wide variety of different things, they're spread out a little bit more, as you might expect. We use a variety of different statistical methods, including machine learning, to try to identify our discriminatory VOCs. In this case, the most important features are highlighted in green. And that's allowed us to identify matrices that really clearly distinguish children who have infection compared to children who are febrile but malaria negative. Just to show you what some of those VOCs look like, this is a promising VOC called non-invasive, which appeared in both our initial study and our validation cohort. Again, probably no one biomarker, right, is going to have sufficient performance characteristics as a diagnostic, but you can see the differences between the children who have symptomatic malaria compared to children who are febrile but negative by other testing. Interestingly, you can see some volatiles that respond to antimalarial treatment. So this, again, is an example of our children who, on day 0, who were positive with falciparum malaria compared to how their volatiles return much to a more normal level after antimalarial treatment. So these studies have given us a lot of hope that ultimately we can identify with larger cohorts some specific biomarkers and combination of biomarkers that might be useful for diagnostics, but of course have raised just about as many questions as they've answered. I get asked all the time, well, what about P-Vivax? Well, we didn't have P-Vivax in our study, so I don't know. What about adults? Well, I don't know because we didn't have those in our study. And then really importantly, when you think about how you might use such a screening test, do our biomarkers appear in individuals who have parasite infection but don't have symptoms? So up to 80% of the people on this planet who have Plasmodium falciparum infection don't know that they have it. And so that reservoir of infection is really important as you think about malaria control. And so fortunately, we've recently been successful with funding from the Bill and Melinda Gates Foundation to actually answer some of these questions. So we have an ongoing study now enrolling children and adults, and specifically looking at individuals with asymptomatic parasitemia and gametocytemia. So our study sites are Zambia and Malawi, partnering again with Carl Seidel and Blantyre and Mabinga in Chipata in Zambia. And we're going to be doing simultaneous enrollment at both sites so that we can essentially do biomarker discovery and validation off of each other in a reasonably powered study. And we're getting breath by multiple different methods. We're also doing skin volatiles, and we'll be phenotyping individuals by parasitemia and gametocytemia. And then we're also getting malaria serologies to try to understand whether any of these biomarkers are different depending on whether or not you have prior malaria infection, which really gets to this question of particularly about children versus adults who may have seen malaria many times. And so we're very pleased to say that enrollment is now begun. So training was completed in February. We've started enrollment in March, and it's going very well. In our initial screening visit, it really highlighted the need for this kind of diagnostic. 35% of the completely healthy children who were screened at our initial screening appointment were positive for falciparum infection. And in the second screening visit, 65% of the children were positive. So these are kids that, as you can see, they're healthy, they're hanging out, they're playing, they look completely fine. And so if we are successful at this, I think it will really be an incredibly useful tool in trying to add to the quiver for malaria control. So as we've continued to work on this work for malaria, it's really like, as I've started to present this, people are like, well, could there be differences in my disease or my infection? And it's really highlighted the need for better pediatric diagnosis for a lot of different conditions. You know, we still stick needles in the backs of babies under 30 days of age who have just a single fever of 38.5, which is seeming a little draconian for 2025. The primary way to diagnose, or the gold standard for how you diagnose metabolic-associated steatotic liver disease is liver biopsy, which, again, it seems like we ought to be able to do a little better than. So we've really launched a number of different studies to do biomarker identification for a number of different diseases. COVID, of course, hit and was immediately of interest. And we were successfully able to partner with the working dog group at Penn Vet. So this has been one of the most fun things that I've been able to be involved with. So Cindy Otto and her team have a group of dogs that are disease detection dogs. So they set them up and they teach them to sniff out odors, and they can present them on this choice wheel in a very sort of scientifically rigorous, defined way. And they kick butt. They can detect ovarian cancer, and they can absolutely sniff out COVID. We gave her samples from children with and without SARS-CoV-2 infection, and they had a sensitivity of 82% and an average specificity of 98%. And I just want to highlight Dixie there. Shout out to Dixie, who literally did not ever get it wrong. Like, unbelievable what these dogs can do. So in case you wondered whether there was real biology there, there's something there. And so we knew even before we did the mass spec that there was going to be distinctions between children with and without SARS-CoV-2, and indeed there are. They're really tremendous biomarkers for SARS-CoV-2, which it turns out we could never get anyone interested in because when we first tried to get funding for this, I was told COVID wasn't going to last. So we didn't need a diagnostic test. And then by the time we had validation, sort of an initial cohort and validation, I was told, well, no one tests for COVID now, so it doesn't matter. But anyway, we have 84% accuracy, 91% sensitivity for SARS-CoV-2 infection in children, just from, this is 4 different breath biomarkers together, that's it. We've also enrolled a group of children with and without the post-coronavirus syndrome, MISC, MIS-C was, at the peak of the pandemic, incredibly difficult to diagnose. We were seeing as many as 2 or 3 children a day at CHOP with this, and they were ending up in the ICU, and children were getting sent home and then coming back in the ICU. It was really horrific. But I think you can see, like, you can see from across the room the breath profile of children with MIS-C compared to children who were completely untreated, who were thought by clinicians in our ER to possibly have MIS-C is completely different. So there's a ton of biology in this breath profile that we see. And so our goal ultimately is some sort of rapid, easy, simple diagnosis for pediatric infectious and maybe even non-infectious diseases. We've got validated biomarkers for malaria, working on asymptomatic infection, proof of concept for SARS-CoV-2 infection. But the challenge that I really continue to have, and this really came up with the MIS-C cases, is that, especially when you've got a disease where the entry into testing is so nonspecific, it's just fever or fever and diarrhea, we're never going to have a clinical cohort that encompasses all the different things it could possibly be. And so just as people say, well, could it be Vivax? Is Vivax going to give the same signal as falciparum? I can't tell because I'm not going to have a cohort that has both of those. I can't tell you whether our breath biomarkers for MIC may or may not be the same as those in cholera or those in cryptosporidial diarrhea because I'm not going to have a cohort that has all of those. And so in order to think about how we're ultimately going to bring these to something that we can use in a real clinical setting, I think we actually have to understand where the breath biomarkers are coming from. So, if I had an RNA-seq signature or RNA signature of disease, you would look at those and say, well, yeah, it makes sense that TNF-alpha is up, or, oh, yeah, it makes sense that CRP is up in this particular condition. And when I show you the breath VOCs, I'm telling you some things go up and some things go down, and I can't tell you whether it passes the sniff test because we don't know where they come from. We have to know where the carbons come from in order to really know what the biological meaning here is. And so that's really the next chapter of what me and my lab have been working on, is really to try to answer this question. And I want to particularly highlight Amalia Berna, who's an analytical chemist in my group, who's really been heavily involved with this work, and then our partner in crime, Andy Cow, who's a collaborator at WashU, who is a microbiome expert. And so we've together worked really hard to develop some tools to simplify the system to really understand the underlying biology of breath VOCs by trying to analyze breath from both mice and babies. So the mice has been hard. This is an obvious, really helpful tool because there are so many different things that you can do to simplify the system in mice, but it took several years to fully figure this out. But we can now tracheally intubate mice, put them on a little mousey ventilator, and collect and analyze their exhaled breath. It takes about a half an hour per mouse, but we get reliably about 200 different volatiles in the mouse. And we can, for instance, tweak volatiles that we give the mouse, in this case isoprene, and really see that appears in the breath. So we have some evidence that the system is dynamic and responsive to the levels of volatiles in the body. And so one of the keys to this has been the use of clean air, or scrubbed air. So you can essentially purchase low VOC air, and that's what we have to ventilate the mice with, because otherwise air itself, just ambient air, has too many volatiles to really see the signal to noise in the mouse system. So that's turned out to be really important to this, to making all of this work. And the kinds of things that we've been doing with the mice, and I'll show you a little bit of this work on the gut microbiome, is both looking at the host side and then the microbiome side of where breath volatiles might come from. So one hypothesis is that host metabolism, right, is important for these breath volatiles, and we've been particularly interested in whether or not the mitochondrial metabolism might be important. And so we've been both analyzing breath from children with primary mitochondrial disease, but also mice that have individual mutations in different components of the different complexes in the mitochondria, and really have some, as you can see, really beautiful signatures of mitochondrial differences in mice. And then we've been working on the gut microbiome. And so the hypothesis here is that microbes themselves make a lot of volatiles, And your body can absorb those just like they might absorb something from your diet or a drug. And they circulate, and anything that's volatile can actually off-gas in your lungs. And so what we've been able to do with Andy Cow's lab is both compare mice that have microbiomes and mice that don't. So germ-free mice, unsurprisingly, have really distinct breath volatiles from mice that have a microbiome. And then we've been able to take mice that have two different kinds of microbiomes. And so the study that's sort of diagrammed there is a study where we take mice who have a microbiome from a lab called Taconic and one from Jackson Labs. They're known to have very different microbiomes. And then we compare the breath of mice that came from those companies, and then take those microbiomes, put them in germ-free mice, and actually look at those conventionalized mice. The output of that is complicated, but ultimately we can actually explain the different variants of specific breath volatiles by the variants in the microbiome. And so that's really allowed us to produce graphs that you see on the right, where we can say that the levels of, for instance, linalool or levomenthol in the breath is highly influenced by the composition and the metabolism that's going on in your gut. We've taken that further to begin to look at specific microbes and what volatiles they produce, and then how that might influence the output in the breath. And so, for instance, here we are putting microbes in a defined culture media and actually analyzing the headspace over individual gut microbes. For instance, E. coli is known to make indole. You can see that in the air. That's in B here. And in C, you can actually compare in a principal components analysis the different volatiles from media, which is in pink, and then 3 different canonical organisms in your gut. And those different organisms obviously are producing themselves different volatiles. But then you can take those organisms, put them individually in the mice, and ask the question, do the things that are enriched in culture, when you culture that microbe, actually also appear in the breath of a mouse that has that microbe? And that's exactly what we find. And so this graph is really summarizing quite a lot of data where we take those individual microbes, put those into mice, and then ask the question, how does that change the breath volatiles of the mouse? And what we see in particular in that upper right-hand corner are breath volatiles that were increased in culture from microbes and also increased in the breath of mice that have been given that microbe, really suggesting that they're actually directly coming from the microbe and making it out into the breath. And then really importantly, we've taken this back to children. And so we were able to enroll a study of children with and without reactive airways disease, asthma. And it's known that children with asthma have very different gut microbiomes. And so in particular, there's a disease-associated microbe, E. cerium. And so you can see on the left there that, and this has been reported by others, the levels of E. cerium are higher in children who are asthmatic compared to children who are healthy. And what we were able to do is, by collecting both breath and stool from the same children, actually look at the components of the breath and develop a model that directly predicts the abundance of E. cerium in the gut of those children. And so we really can demonstrate that even in children, we're getting direct information about what's going on in the gut from what we see in the breath in a non-invasive way. We've begun to try to take this to babies, and the reason we're interested in babies is that they have themselves a reduced component of microbes when they're first born. And so obviously you're born with few gut microbes, and you acquire those during your early postnatal life, and then they mature significantly as you change your diet, as children stop breastfeeding or nursing, and then as they begin solid foods. And so we had some challenges in developing a system to collect breath from neonates. We had to go, as you can see here, to the SIM Center at CHOP because the IRB wanted us to prove we were not going to suffocate the babies with the one-way valves, and so we had to actually measure the exhaled pressure that it was required to fill the bag, et cetera. Anyway, we proved to them it was all safe. Everyone was very engaged with it, but they didn't want us to suffocate babies. But we did not. And so now we've begun to collect and analyze breath from newborns. So we can collect on day 1 of life, newborn breath. And you can see an increase in, again, our sort of canonical breath volatile isoprene. But you can also see that newborns are going to be a great system for reducing the complexity of all of this, because they do also have fewer breath volatiles than adults do, or school-age children. So these are samples collected exactly the same way, exactly the same volume from infants and from school-age children. And features here is roughly correlated with the number of compounds that are present. And so it just reduces the complexity of all the math to have fewer things to compare. So fewer breath volatiles, fewer microbes in the gut. And so we're sort of off to the races to figuring out some of these things. And so we have 2 ongoing clinical studies that are currently enrolling. One that's enrolling well newborns. So actually, we've already completed initial enrollment of 50 infants enrolled within the first 48 hours of life. Most of those have gone through their first 6-month samples. We've started to get the 18-month samples. 18-month-olds are pretty angry about the whole breath collection thing. But we're pushing through, and on all those kids, we're getting breath VOCs and fecal samples at the same time points. And then we're sort of midway through enrolling a set of infants where we're actually trying to look to see how the breath volatile profile changes with antibiotic use. And so many infants who are seen in our emergency room with fever in the first 30 days of life are going to end up getting antibiotics. And so we're collecting breath and fecal samples before they get their antibiotics, and then the next day after, to see how whether or not your breath changes essentially quickly when the gut microbe components change. So more to come on both of those. So our vision for the future really is to put a name to where each of the volatiles that we've been looking at come from. So that, there's 200 to 400 different things. So we've got, a little bit of work cut out for us. I'm sure some of them are of dietary origin, and we have some ideas on that already. And we are hoping to begin to classify things on host metabolism, both the mitochondria. We have a population of children with inborn errors of metabolism at CHOP that we're also beginning to enroll, and then understanding how the gut microbe both impacts the dietary origin things coming from the host, how the gut and the host work together. I think it's all going to be pretty complex, but I'm hoping that we can learn a little bit about what information we're getting from the breath in our patients. I just want to give a highlight before I give my acknowledgments, one note of gratitude for the federal funding that was involved with both my own training and the work I present here. So, I was T32 funded as an MD PhD student. I was GME funded as a resident, T32 funded again as an ID fellow, had a career development award, and our very first foray into breath was an R21, and has had subsequent awards, but really all of it depended on federal funding to happen in the first place. So, shout out to that, and long may it continue for others. And obviously, I want to acknowledge the folks in my lab. They have a TikTok. I was not responsible for the name of the TikTok or its content, but I do like the little intro video, which is hysterical. And many thanks to my current funding sources, our collaborators, and our colleagues, and our patients and families. So thank you.
Like on the shape, and it's just the fight. That was amazing. Really neat. So one of my questions is, is the volatome like a local thing? If you measure people here and you go halfway around the world and you measure like the same age kids, do they have some things that are different, but some things you can count on being the same?
Do I need to repeat the question for the folks online? Yeah. So the question was about how the breath volatile might change depending on your geographic location. We think that our evidence is that a lot of it is the same. There are definitely some things that are different. One of the things we do to control for that is we try to collect a room air sample every single day, and we only sample in the same room in a place. So for instance, in Malawi, we have a room that's used for the breath collection that therefore is not like got a lot of through traffic, et cetera, so that it's not got a lot of day-to-day variability in the same way. And then that allows us to normalize and reduce the number of things that are just ambient contaminants, essentially, which definitely have some geographic changes. But the overall component, like what are those, say, 400 things that are commonly in breath, they're pretty much the same 400 things.
Yeah, that was a great talk. Thank you very much. Before you sample the breath, do you have to like do some sort of mouthwash? Like if I have dog breath, or does that get filtered out? Like, can you filter out my dog breath?
So the question is whether or not you need to do a mouthwash or something before you collect breath. We do try to avoid the upper airway, essentially air. So in adults, you can coach people to kind of out that upper airway, and then you're really trying to collect the alveolar breath. You know, there's a little variability with whether you can do that with a child. So the 3-year-old's not so good at that. An 8-year-old is usually pretty good. So we try to enrich for the alveolar breath. Obviously, the babies, we just get what we get. But it's part of the reason that with the mice, we have to tracheally intubate them because they're coprophagic. And otherwise, we just get their literally poo breath, and that is not so useful.
Audrey, really nice to see your progress over the years. I'm wondering if with your kids in Malawi, you know which ones like have active TB and if there's a way to use some of your volatiles to help diagnose today.
The question, that's a great question. So the question was about using TB to diagnose, using breath to diagnose TB. So of the diseases where a breath has been tried, TB is probably the one where it's been the most successful. So there's been a number of studies looking at volatiles for TB diagnosis, both in adults and pediatrics, and it actually is terrific. So there's some really great breath biomarkers for TB. And I was at a meeting that the Gates Foundation was for breath-based diagnostics. They have an entire TB program. So that, I think, actually has a ton of hope.
Will it be commercialized?
I certainly hope that it will be commercialized, yeah. Yes.
So 2 questions. This was a great talk and it's great work. Thank you so much for coming to present it. One of the questions is whether you've done anything longitudinal to track what happens, the volatiles over puberty. And I'm thinking those about a pubertal transition and sex differentiation in the volatile.
Yeah, great question. So the question was about sex differentiation or the effects of puberty on volatiles. So we started to do that a little bit with some skin stuff because we were really pretty sure there were going to be skin volatile changes in puberty. And we have an ongoing study on skin volatiles that will include that as one of the variables. We haven't done that longitudinally with children yet. This study of the babies is the first longitudinal, like long-term study that we've done. But I think that's it's a really interesting and important question.
For the malaria part of the talk, have you been able to look at individuals who are infected and symptomatic, infected asymptomatic, and then infected treated as to how this may change and whether you can use it to track the effectiveness or lack of effectiveness of any anti-malarial therapy?
Yeah, so that's exactly what we had hoped to do with that second study. We had hoped that some of those healthy children that were healthy siblings that were enrolled would end up being asymptomatically infected. And interestingly enough, hardly any of them were. So we only ended up with like 2 asymptomatically infected kids in that cohort. So we weren't able to answer that question. But we can definitely see volatiles shift as children get treated. So I do think there's information there about the effectiveness of treatment. Yes.
So I enjoyed your talk very much. I see one of the biggest challenges is the complexity of molecules and deconvoluting the patterns that you want out of it. Have you thought about trying to use AI or machine learning to find hidden patterns between different metabolites and maybe its specific relationships that might be diagnostic for specific conditions?
That's exactly how we do it, yeah. So the question was about how we find the information from this complex mixture of volatiles, and we do AI and machine learning techniques are exactly one of the tools that we're using. And also trying to, in some of our, especially the cohorts of diseases where we're looking in the US and have all the clinical metadata. So it may be, combinations of, this clinical lab plus these volatiles might be what gives us the signal that we need. Yes.
So this is a very cool talk. It was mostly focused on the biomarker discovery side, but do you have any ideas for what the final sensor is going to look like? The GSCAMS is pretty bulky, like, so is it got to be the same kind of technology? Something else?
Yeah, so the question is actually how you operationalize this, how you go from biomarker to something that would be a point of care test. So this is an active area of engineering interest. So and again, the Gates is actually funding 5 different groups right now working on these kind of problems. But among the things that are commonly thought of as the sort of the translational pipeline for this are sensor arrays, where essentially you have different sensors that are like metal that will interact with volatiles, and the circuit will change depending on whether or not the volatile is bound. And then you can even add in little heaters that change that essentially clean the sensor once everything has been bound and interacted. So those kinds of sensor arrays are actually apparently pretty cheap to make and pretty like heat and humidity stable. And so the idea is to ultimately get down to a sensor array that would be in the couple $100 cost, which is very expensive for these low middle income settings. But at the same time, if you could use one device for hundreds, thousands, whatever of tests, then I think it becomes cost effective.
Kind of pulling across the full talk, you started with the in vitro, like VOCs of malaria, and then at the end, looking at the bacteria. I was curious, in malaria, how much of those biomarker VOCs are overlap with in vitro malaria culture? Like, are those host-derived or are those...
Yeah, so the question is overlap between what we see in vitro and in vivo in Plasmodium infection, and we do see overlap, but now that we've done more cohorts of different diseases, I actually think that some of what we see in the breath of kids with malaria is that actually we do see in culture in that child is not actually coming from the parasite. So I actually think that, again, with this microbiome work, some of what we're seeing in the breath is actually a sign of gut barrier dysfunction in the kids with gut sequestration. So we're trying to begin to ask that question specifically, because what I think we're seeing is an increase in these microbe-generated VOCs that are appearing in the breath.
Kind of a follow-up on that. I mean, for the dog study and for a lot of these studies, the comparison is a healthy control. And so do you think it's possible to get to any level of specificity, or is it going to be a sick versus not sick?
Yeah, the question is about specificity, which is the question, right? So in the healthy versus SARS-CoV-2, we were stuck at the time that we did that pilot cohort because we weren't allowed to send kids into rooms and with individuals who had respiratory illness that weren't phenotyped. When we have subsequently gone and looked at just acute respiratory illness in children, viral illness of other sorts, it's not specific to SARS-CoV-2. It's probably not surprising to people that it's not, but I'd be interested in the FDA-approved study, if it is, because ours certainly were not. And so, I think those breath biomarkers are sick versus not sick. And again, it gets down to, well, where are they coming from then? And also, for instance, would it be the same if the child were neutropenic? Would it be the same in a child who has an immune system? So are they coming from the host immune system? Is that important for where they're coming from? All of those things. And I think if we knew that at the get-go, we would have gone in different directions. Yeah.
How dispensable is terpene production? Like, are we at risk for selecting against it diagnostically like we are with HRP2?
Great question. So the question was, how dispensable is terpene production? Well, I think that it is not dispensable because the enzyme that we think is making them in the parasite is essentially a co-opted enzyme that's an essential enzyme for other metabolic pathways in the parasite. So without getting in the weeds too much, I don't think that the parasite can get rid of that enzyme. Yes?
I was wondering about genetic variation between different individuals, and if you've had a chance to look at the mothers of the babies
that you're looking at?
Oh, great question about genetic contribution. So this is part of the reason that we are interested in doing the inborn errors of metabolism kids, because that'll be sort of the extreme version of that. We really expect that we will see big signals there. There has been a study in Europe looking at genetic variation. So isoprene, as I mentioned, is a really dominant signal, and you can actually find individuals that have very low isoprene levels. And they did a GWAS study and actually identified the genetic change that gave them low isoprene.
And is there any variation with the malaria belt versus non-malaria? Have we selected?
I don't think we've not done those studies well enough to know, but that's a really interesting question.
Kind of a follow-up on that is we have a bunch of online questions. We're gonna not go through all of them, but one of them is people with hemoglobin variants are protected from malaria, and whether there's a difference with the variant.
I have not. So the question was about hemoglobinopathies and whether that might impact volatiles. We have not looked. That would be an interesting question.
Great. Well, thank you, everyone. And thank very much. Thank you.